more work. So stay tuned.
Thank you very much, Trevor. I see the first question. Tom.
Thanks for the talk, Trevor. Your correction factors for the callback library divergence. Does that require some special assumption, or is this true for any callback library divergence between any two distributions?
So it's assuming the words, when you put the words into blocks, it's assuming that they're individual. So there's no correlations between words. But we would expect this to be true when the words are large enough, because the correlations between the words should be sub-extensive to the length of the word itself. So as you systematically increase the words, the words will effectively become independent.
Okay. All right.
David is next.
Okay.
Hi, Trevor. Very nice talk. Very, very cleanly presented. A related question to Tom's. Even if you're just estimating samples of an IID process, estimating the bias of an IID process from samples, there's things like the Laplace sample size correction, and more generally, the quote-unquote proper way would be to do a Bayesian estimate. So you have to specify priors. There's been a lot of work extending that to estimating entropies and arbitrary information theoretic quantities from IID samples of anything, based upon things like Chinese restaurant processes, Dirichlet processes, and so on and so forth. Have you tried any of those? Because one can get much more sophisticated, especially when you're trying to estimate something like a function of a distribution from the distribution, which is what you're trying to do.
Yeah. So the short answer is no, I haven't tried that, but I think this is the direction that I'm hoping to sort of take with this project.
In particular, there's been some work where it you do not assume you actually even know the size of the state space. So your prior extends over all state space sizes, and that seems like it might be quite relevant in this domain.
Okay, yeah, thank you. I'll have to.
We can follow up offline on that.
Next question by Ivan Garashuk.
Hello, can you hear me?
Yes.
Yeah, okay, thank you, perfect. First of all, thank you for the talk, and second, my question might be a little bit, naive, but how is, at the very end, you showed that the memory effects range from somewhere of like 40 milliseconds to 200 milliseconds. Here from your data, how are they related to the refractory periods of said like neural data? Because I would assume, like, even if each individual neurons does not have any memory effects except for the refractory period after the action potential, you would get probably similar effects. I think.
That's a good point. So I think this is supposed to be longer than that. So you're trying to measure correlations between spikes as well. So like if the words are long enough, the idea is you're trying to get how correlations between spikes gives, how is that different than a process with no memory? So so the idea is, you're trying to look at much longer than that time scale.
Yeah, thank you.
Okay, thanks. I see no more questions, so let's now move to the first invited talk. This is Sreekanth Manikandan from Stockholm.
Yeah, hi.
Yeah, so we can see you. You start sharing screen. Perfect.
Yeah. Thank you. Thank you for the invitation to this program. I'm Sreekanth Manikandan. I'm a researcher in the soft matter lab at Gothenburg University now. I was previously a PhD student at Stockholm University, a postdoc at Nordita in Stockholm for 2 years, and then also a postdoctoral scholar at Stanford University. So I'm going to talk about localizing entropy production in cellular processes. It is going to be related to some of the talks that we had today. So let me start with cartoon of a cellular process. Here I have cell division, which according to me now is one of the most beautiful processes within a cell. And you see that when a cell is dividing, it is using its machinery to separate out the genetic material into the two daughter cells. And this happens millions of times in our body. And living systems execute processes such as cell division with high precision and balance in fluctuating environments. And now these are not free, but they are powered by molecular motors that break down simple sugar molecules such as glucose. And as usual machines, some of this energy is utilized for cellular function, but most of it is dissipated as heat to the surroundings because all these functions happen in fluidic environments and also as matter. So over the years, we have made significant progress in visualizing cellular processes. We have a detailed understanding of the underlying biochemistry. As the first speaker mentioned today, we know the entire biometabolic pathways. But as we know, as we discussed today earlier, there is one significant open problem, which is that we lack accessible methods to locally, spatiotemporally quantify energetics of nanoscale cellular processes. Now, why is this technologically a problem? The issue is that if you look at ATP hydrolysis, which is, let's say there's an event where 1 ATP hydrolysis is taking per second, that would correspond to about 10 to the power minus 7 picowatt of energy. Now, if you look at the state of the art calorimeter that is available, that has a resolution of about 26 picowatt. Of course, there is some ambiguity about what is the best calorimeter in this context, but this is the information I found. So there is at least 7 to 8 orders of magnitude difference between the current technological sensitivity that we have versus what we need to be able to measure a single biomolecular event within a cell. Now, over the years, stochastic thermodynamics has helped us make significant progress in this direction, I would say. So first, let's just focus on energetics at kBt scale. So we know that for analytically tractable systems, such as a colloidal particle in a harmonic trap, where all the conservative and non-conservative forces acting on the system, there is an equation describing what is the heat dissipated along a single trajectory. Now, for the system, for a typical colloidal system, you have forces of the order of piconewtons, and the particle moves distances of the order of nanometers. So naturally, you end up with regimes where you have energetics of the order of kBt. But as you know, not all systems have such underlying exact analytical descriptions. No. So let's say, what is the next best thing that has come out of stochastic thermodynamics? So stochastic thermodynamics has provided for control settings another bunch of approaches for quantifying energetics, generically speaking. So for example, fluctuation theorems. We have used fluctuation theorems combined with single molecule experiments to show that breaking a single base pair of RNA hairpin would lead to 10 to 12 kBt of energy. This thing that I'm not going to have a complete review here. This is just some papers that come to the back of my mind quickly when I think about this issue. So then there was this Harada-Sosa inequality, which was proposed in 2005. And in 2018, there was a study where they used it with kinesin motors under load in a reconstituted setting. And it has been shown that the dissipation is about 15 kBt per second in kinesin in reconstituted systems. Now, there has been a lot of studies, but going fast forward, now we recently had variant sum rule, which was applied to RBC flickering data. And then this method showed that if you account for, scale the total entropy production with the area of the cell, you get amounts about 10 to the power 6 kBt per second, which is compatible with the estimates that you have from calorimetric, other calorimetric measurements, because you're reaching that regime now. So this is the second sort of methods. Now, what is the third approach? This is what I'm going to be mostly interested in, which is, can we have minimally invasive methods? What if you don't want to put a probe into your system? What if you don't want to perturb your system? Can there be minimally invasive methods of inferring energetics or dissipation in cellular processes? So that is the inference problem. Can we non-invasively obtain energy dissipation and dissipative force fields directly from experimental time series or movies? So if you think about time series, as just Trevor talked about in the previous talk, there has been, Edgar and collaborators have pioneered in this approach of measuring Kullback-Leibler-based reversibility from trajectories. And then, of course, through stochastic thermodynamics, there is a direct connection between this reversibility estimate and entropy production. And recently it is shown for fluctuations of hair cell bundle oscillations, where also combined with the TUR, you get up to 10 to the power 3 kb per second in entropy production. The other approach which I particularly like and I have taken a lot of inspiration from is stochastic force inference methods directly on trajectories as well as Brownian movies. And more recently, we are also starting to see a lot of developments in using machine learning based methods to directly infer entropy production from time series data. So I'm going to talk about an approach which in the development has taken a lot of inspiration from these techniques, but it is based on the thermodynamic uncertainty relation. So thermodynamic uncertainty relation was proposed in 2015 when I started my PhD, and it was proved later in 2016. And as you see here, thermodynamic uncertainty relation is a fundamental precision dissipation trade-off for non-equilibrium systems. So it states that if you can measure fluctuations of currents, here J are currents in a non-equilibrium steady state, and tau is the time duration for which you measure the current. And if you measure a statistical quantity as the mean square or variance of this current scaled with the time duration, you get an estimate of precision of this process. That gives you a lower bound to dissipation. That is the idea. And naturally, following this, there has been attempts to find out what is the best current, which would give you the best possible estimate for dissipation. One paper I particularly like is this Lee et al. And from 2019. So then initially saturation limit in TUR used to be this equilibrium limit where sigma itself is vanishing, and then you need a very long time to have sufficiently large current outputs in this case. So this large time limits have a lot of mathematical background in it, because you have large deviation theoretical structure giving you proofs and analytical support for results in this regime. So during my PhD, we had a slightly different approach, which was to study non-equilibrium systems as a function of time. So this was done using a path integral based approach. I won't go into the details, but let me show you what we get when you do this when you apply this to TUR. So here we have a system where we are studying TUR as a function of time. The system has a true entropy production rate of 2 kb per second. And here blue and red lines are estimates of entropy production obtained by using TUR as a function of time. So as you see, at a large time, almost all the currents for different currents. So here, for all currents at a large time, the inference predicts a value which is roughly 3/4 of the true value. Now, as you go shorter and shorter in time, there is a lot of variability. But in particular, there is one current which is saturating the TUR. So we were able to show that actually for overdamped diffusive systems, if you take the tau going to zero limit, the thermodynamic uncertainty relation saturates. And not only that, the current which saturates the TUR is nothing but the fluctuating entropy production. So you can essentially write it in terms of this conjugate force field, which is called the thermodynamic force field. I will just call it the dissipative force field in this talk. And you can obtain it in terms of that. So almost simultaneously, this was also obtained by two other groups, one by Tanwan Wu and Hasegawa, and also Shunotsubo, Takahiro, and others. So there is something I like about Tanwan Wu's proof, which is that there is an analytical solution to this optimization problem given a finite set of basis representation for the currents. And then the interesting part about Shunotsubo's work was that it provides you a machine learning based framework for optimizing TUR in the short time limit. And they also showed that the saturation is independent of the initial condition, initial condition of the first point when you have the trajectory. So let me show you, let me just present the scheme. using the approach that Shun and Takahiro presented. So I'll call it the short-time inference scheme. The idea is that you get an ensemble of short trajectories from your system. You assume there is no hidden degree of freedom. You're measuring all phase-space degrees, and it is an overdamped diffusive process. Then you model your unknown dissipative force field using a neural network. And then with this neural network, then you can construct currents, single step currents, so that you are in this short time regime. And then basically you want to train this model function to better approximate the dissipative force field. And the cost function for the training is given by nothing but the thermodynamic uncertainty relation. So what the proof of saturation in the short time says is that if you work with sufficient amount of data, and if you are working in the sufficiently short time regimes, the thermodynamic uncertainty relation is guaranteed to saturate, and you will have an exact estimate of the dissipation rate as well as the dissipative force field in this way. And since the initial condition, the proof of saturation of at the short time is independent of the initial condition, what you get is that this method automatically generalizes to time dependently driven scenarios. Okay, so here I have, so in the following, what I will do is I'll show you an experimental benchmark, an application to some numerical simulation, and then an application to a biological problem. So here we have results from an experimental benchmark where we looked at the system where you have a colloidal particle trapped in an optical tweezer. So one of the degrees of freedom is the position of this particle, which is x here. Then you are driving the system by shaking the minimum of the trap using a non-Einstein Olympic process. So you have the second degree of freedom is essentially the center of the position driven according to a non-Einstein Olympic process. So this is a linear Langevin system which you can analytically solve, and then you know analytically what is the expected entropy production rate. Indeed, when you apply this short-time inference scheme on this data, you will get we got estimates which are in perfect agreement with with experimental errors, good agreement with the theoretical predictions. So here the parameter theta corresponds to the amplitude with which you are shaking the trap. So higher the theta, you are shaking the particle more vigorously, and the system is more out of equilibrium. And so here there are various theoretical and experimental error bars put, but also an agreement with the previously known inference scheme, which is this stochastic force inference scheme. And then you see that TUR as well as the stochastic force inference gives exactly the same value for entropy production for the given data set. Now, this method, as I said, also reproduces the dissipative force fields. So on the left-hand side, you have the dissipative force fields inferred from the data, and on the right side, which is the force field that you know from theory, and there is agreement to both nanometer scales and piconewton magnitudes. So what you could do next is actually use the knowledge of this dissipative force field. You have the trajectory, and then you can essentially color the trajectory with entropy production locally in space and time. So that is what I show in this plot here. So here I have the same trajectory data as before, but now it is colored with the fluctuating entropy production, which I learned from the same trajectory. So in this sense, it's very powerful that you basically just need trajectory data, and TUR gives you a model independent way of identifying fluctuating entropy production along the same trajectory data. So now this is, so far I showed you this for a linear system. If we have a nonlinear system, this should also work. So here we have an example where I show you how the learning works online. So I have a system which is a particle in a double well potential, and it is acted upon by two temperatures in orthogonal direction. There is a hot temperature acting on the x-axis, and then a cold temperature for the particle on the y-axis. It's called an anharmonic Brownian gyrator. So here, what you see is essentially on the left-hand side is the trajectory data, and in the center is the entropy production as well as the dissipative force field inferred as you get more and more trajectory data. And you see that there is a highly non-trivial dissipative force field as well as local dissipation structure that is emerging, which is, this is how it looks at the end of the inference algorithm. But you are again getting it straightforwardly from the trajectory data. Now we could apply this to time-dependent cases. So a classic problem is probably bit erasure. So here you have a bit of information which is equally populated in an asymmetric double well initially, and then at the end of the protocol, you are getting the information to be raised to one of the wells. And one can again apply this algorithm and then identify which part of this process is the system more irreversible. So here is a color map now on the same trajectories, which is showing you that certain regions of the process, for example, when the potential is not so much changing, we expect the system to stay close to equilibrium, which is coming out. And when there is rapid changes in the potential, there is regions of high entropy production. I mean, this is expected, but here you get a model independent estimate of this quantity from the data. Okay, so the original motivation was now apply to try to see if these things work in a biological experimental context. So to this end, what we looked at is the system of cell membrane. So cell membrane is, as we know, one of the most dynamic and integral part of a cell. It plays a crucial role in signal transduction, cell-to-cell signaling, cellular uptake, and much more. And the interesting part is the role of various biomolecules in the dynamics is known. So we know what enzyme or protein to knock off so that the membrane does something else which it was not doing before. So these experiments were carried out in the lab of Bidisha in IISER, Kolkata. So what you see here is the basal membrane fluctuations of a cell. So here a HeLa cell, which is attached to a substrate, it's an adherent cells. And what you're viewing is the part of the membrane which is attached to the substrate. And the pixel intensity here essentially corresponds to the height of the membrane at that point. So, and it's flickering because there is a membrane dynamics. And so if I look at a patch in this membrane, let's say this region, which is in blue color, I can follow it in space and time. Each patch is made up of several pixels. And then one can, if I look at the fluctuations in this over this patch or of a pixel over time, you see that there is roughly 40 to 60 nanometer height fluctuations. Now, this is how a typical inference scheme looks like. This is inspired from the work of Agnesetto and Brownian movies and stochastic force inference. So you take a movie of cell membrane dynamics, and then one applies for each patch that I saw, that blue square, I apply a dimensional reduction so as to actually get rid of the noisy part of my data. So you can, as Trevor mentioned in the previous talk, you can shuffle the data and find out where is your noise level and pick out components which are above the noise level. And then you apply this inference algorithm on the data. So each patch here would now correspond to a color which corresponds to dissipation that I inferred using the short-time inference scheme. So this is how the control case looks like. So here on the left-hand side, you have two different cells. And at t equal to 0, you make a movie of about 10,000 frames at a rate of 50 millisecond. And then each patch, you pass it through the inference algorithm, and then you get a number. You color the cell according to this number. So as you see that clearly the cell lights up, that there is high red color here corresponds to high dissipation and blue corresponds to low dissipation. So there is high dissipation over the surface of the cell, and in the background you see low dissipation. And then if you do this following experiment where you ATP deplete the cell, so you treat it with a drug that essentially depletes it of ATP, then you see that this color goes down. So there are the regions which were initially high in red now starts becoming more and more blue. And one can verify this over a population of, we did up to 30 or more cells. And then you see that always there is this reduction in the heat dissipated, or I mean, it's actually not the full heat dissipated, but the estimate we obtained using TUR is going down, which is consistent with the fact that when you do ATP depletion, you expect dissipation to go down. So another experiment that we did is where you treat the cell with another drug called cytochalcin D, where you essentially weaken the cortex of the cell. And in this case also, we obtained not as profound, but a small reduction in the dissipation estimates from the cell membrane fluctuations. So yeah, with that, I go to the concluding part of my talk. So here, what this method is able to do is to spatiotemporally resolve energy dissipation and microscopic forces in minimally invasive ways. We haven't had to put a probe to the cell in this case. It is applicable to both time-lapse movies of fluorescent label processes or natural contrast movies. And since you can work with machine learning type algorithms, it's straightforwardly generalized high dimensions, and also, as I showed, to time dependently driven cases. But of course, you need a lot of data in these cases. But it is limited by, but consistent to the degree of coarse graining. Limited in the sense you will get a lower bound when you have a coarse grained system using this method. But for example, the cell membrane. But what I mean by consistent is that we looked at this particular example where you have now not one particle, but two particles in adjacent traps, and their positions are given by x1 and x2. And then one of the particles you are driving with an Ornstein-Uhlenbeck process, as I showed before. And then in this case, I can compute what is the total entropy production in the space of lambda x1 and x2. And then I can also ask the question, what is the entropy production in this coarse-grained phase space of just x1 and x2? What we found is that, so as you expect in the total phase space, you get perfect agreement with TUR-based inference and the theoretical value. And for the coarse-grained phase space, recently there are results about how you compute this coarse-grained entropy production. And there is also a perfect agreement in the coarse-grained phase space when you work with the TUR-based estimate. So I think there is also a lot of possibilities in understanding coarse-grained entropy production using TUR-based approaches. Yeah, so to end my talk, I stated an open problem. It's a interesting open problem, I believe, and I believe going forward, a combination of model-based predictions and model-independent inference, such as the one using TUR, and a new generation of quantitative experiment, particularly probing biomolecular level processes, could help us reach energetics of cellular processes much before a calorimeter is built to do the same task. Yeah, with that, I would like to also thank my collaborators. So Supriya was my PhD supervisor, and much of this work is done in the labs of Ayan Banerjee and Bidisha in IISER Kolkata, India, by students Bishajit, Tanmay, Avijit, Shubhra Kohli, and Shun and Takahiru was part of the time-dependent inference problem. And also thanks to Grant for various discussions on this problem in the later stages. Yeah, thank you, and let me know if you have any questions.